I'm going to go ahead and get started for the sake of time. I'm going to read you the first question, and I'm going to give you a couple of seconds to solve it yourself, and then we're going to solve it together. Okay? So the first one says, the diagram shows the elements present in two organic molecules, W and X. Which molecule could be W and X? Which molecules could W and X be? So we have molecule W on the left-hand side, molecule X on the right-hand side, and it shows that there's phosphorus just in molecule W, nitrogen in molecule X, and then between both of those, they have a commonality of hydrogen, carbon, and oxygen. What are you guys thinking? Someone says it's this HL or SL. This is SL, but you can also do these questions if you're HL, right? Because most of them are also on the HL paper. This is a specimen paper. Okay, we have a C. Someone's saying C in the comments. C. Good guesses, guys. So what made you guys think it was C? Hopefully the phosphorus gave it away, right? Because monosaccharides are just carbohydrates. We know carbohydrates do not have phosphorus. So we can cross out A. Nucleic acid. It does have a phosphate group, right? So we'll leave that option. Triglyceride does not. But what do we see on molecule X? We see that there's nitrogen. Do fatty acids or glycerols, triglycerides, do they have nitrogen? They don't, right? So we can cross out B, and therefore our answer is C. Did this make sense, everyone? Hopefully it did. It's a pretty solid first question. It says, what is the consequence of the ability of water to form many intermolecular hydrogen bonds? What are you guys thinking for this one? So intermolecular hydrogen bonds, we have to think of the properties that intermolecular bonds provide us. So we have a C in the comments. Just going to make sure I didn't miss any questions. Phosphorus definitely gave it away. Good. I'm literally just watching your past videos on my crash course. Yay! Good. Okay, we have 2 C's, 3 C's. Very good, guys. You guys are answering very well. C. Okay, let's go through the answer options in case someone was a bit not okay with the question. We're going to read it together. So it says, ice has a higher density than water and floats, providing habitats for fish-eating mammals. Does ice have a higher density than water, or is it a lower density? We know that ice has a lower density, and that's what actually allows it to float. So that's why we can cross out A. Next, it says light can pass through water, so algae can photosynthesize. That is a true fact, and it's actually very beneficial for our environment. But is that a consequence of hydrogen bonding? We know that that is not a consequence of hydrogen bonding. That's why we can cross out E. Next, it says some small invertebrates can walk on water surfaces. That reminds us of what properties of water. Can anyone name them? They start with C and A. Cohesion. And what's the second one? So this one is definitely our answer. So let's read D just to double check. It says small amounts of energy are needed to change water from one state to another so that evaporation rates and condensation rates are rapid. It's not small amounts of energy, it's large amounts of energy. Remember, water has high latent heat of vaporization and high specific heat capacity. Very good, guys. You guys are saying cohesion and adhesion. Perfect. And that's why we know our answer cannot be B. Yeah, so cohesion and adhesion are the properties that hydrogen bonds allow us to have, and that's why invertebrates can walk on water surfaces. Okay, perfect. We move on to question 3. It says, which molecules are produced during the hydrolysis of triglyceride molecule? So what makes up triglyceride molecules? Can anyone tell me in the chat? Let's see. So triglyceride, we know it's part of the lipids unit. Someone said A, water and glycerol. Does water make up our triglyceride? Or is it something else? You got the glycerol part correct. Someone said C. So hydrolysis of triglyceride molecule. We have B's, C's. Okay, I'm seeing a lot of B's. Very good, guys. It is B. So when we hydrolyze a triglyceride, hydrolysis means breaking it up. When we break up a triglyceride, we're left with a glycerol molecule, which is usually drawn like this, and 3 fatty acid chains. Maybe you've seen something similar like this, but you've seen it with the carbon and hydrogen and the actual specific elements. We can simplify the drawing by drawing it like this. We know that it's one glycerol molecule and 3 fatty acid chains. So when I break up my triglyceride molecule, I'm going to be left with glycerol and fatty acids. So you could think of this as, how am I going to make up a triglyceride molecule, and then work backwards as what are the products when I break it up. Okay? Hopefully this question made sense. Just let me know if you want to go over it again. It says the diagram shows the structure of leucine, an essential amino acid. Which diagram highlights the part of leucine that distinguishes it from another amino acid? So which part allows us to differentiate this specific amino acid from other amino acids? I'll give you a couple seconds to think about it. I know a lot of students usually hate the diagram questions. I'm just going to zoom in so you can see, but try to reflect. What's going to help us differentiate? Someone said, sorry, I can ask if you're going to go only because I've solved this paper. I am. Today I'm only going to go for paper 1A, but if you're interested in paper 1B, please let us know so we can maybe do another live session tomorrow. So you have A's and B's. Okay, what are you guys looking for specifically? So very good. Someone said B because it's the R group. Good. So the thing that differentiates each amino acid is the R group. Usually the structure of the amino acid is a amino group, amino, a carboxyl group, a hydrogen attached, and then the specific variable R group. So this R group is what distinguishes specifically different amino acids. So what are we going to circle in our answer? We're going to choose the answer that circles the R group, right? So let's have a look. We can see here that option C shows our carboxyl, our amine, and our H. And we just said that this is the same specific functional groups that are in all of our amino acids. That's why we're going to cross out C. Let's have a look here. In A, it does circle some sort of group. Okay, let's have a look here, because we see that we have our carboxyl group here. We have our hydrogen here, and we have our amino group here. So this, and this. But we see that it doesn't fully circle the R group, and that's why A is wrong. Let's take a look at B. In B, we have the carboxyl group, the amine group, the hydrogen group, and then we can see it circles perfectly the R group, and that's why our answer is actually going to be B. Let's have a look at D quickly. We see our carboxyl group, our amine group, and our hydrogen, and it does not circle our R group. That's why D is incorrect. Does this make sense, everyone? And not all of R group is selected. Very good. I'm biochem grad student. Ignore my first mistake. Okay, good. So let's do the next one. It says, what is a common feature of enzymes? So think about enzymes, their type of proteins, right? Globular proteins. Is it A, they all react with substrates? They all decrease the rate of reaction. They are all secreted from cells and vesicles. They all bind to the active site of the substrate. What are we thinking? I'm going to change the... Hopefully that's a bit better. Okay, people are saying D. Be very careful, guys. Reread the wording of D. They all bind to the active sites of their substrate. Do enzymes bind to the active site of their substrate? Or is it the other way around? Okay, lots of people are saying A. Very good. Exactly. So the answer is A because they all react with substrates. Okay, so enzymes here, this is like an example of a globular enzyme, just a quick drawing. We have an active site where the substrate binds, and the substrate, this is our substrate, it comes and binds here. So yes, all enzymes react with substrates. We can say that that's correct. Okay, they all decrease the rate of reaction. Do they decrease or is it they increase the rate of reaction? They increase the rate of reaction. So actually, B is incorrect. They're all secreted from cells and vesicles. That's not true because some enzymes stay inside the cell. So we cannot say they're all secreted. That's why we can cross out C. And then they say, D says they all bind to the active sites of their substrate. That's not true. Enzymes do not bind to the active sites of their substrate. Substrates bind to the active sites of enzymes. So we can cross out D. Hopefully that makes sense. They increase, yeah, look good. Yep, good job, guys. Something because it increased the good. Whatever you guys can. So we do have a crash course if you're interested in joining that. Okay, I think everyone understood this one. We're going to move on. It says, what distinguishes aerobic respiration from anaerobic? Can you go with number 4 one more time, please? I can if you'd like. Number 4. Okay. People are saying D. I'm just going to quickly go over 4. So the reason why 4 is B is because we have to circle the variable R group. The R group is what distinguishes specific amino acids. We know that there's a carboxyl functional group that's common to all amino acids. We know there's an amino group that's common to all amino acids, and there's a hydrogen, which is common to all amino acids. The only thing that is different, the distinguishing feature between different amino acid is the R group. So you just have to circle the R group, and B is the only one that shows us the R group. Hopefully that made sense. Let me know if you want me to explain it again, and if so, I can come back to it at the end. Okay, what did we say for this? We're seeing lots of D's. D, yalla. Good job. A, D. Okay, good job, guys. So only aerobic respiration produces carbon dioxide. Very good. D is the correct answer. So aerobic respiration is respiration with enough oxygen. Anaerobic is with limiting oxygen. So there's not enough oxygen here, right, to produce ATP, and that's why it's called anaerobic respiration. Let's have a look at the answer options. So A says mitochondria are only used in anaerobic respiration. Not true, they're used in both. Aerobic respiration yields lower quantities of ATP. That's not true. It yields higher quantities, a lot more actually. And then only aerobic respiration can use glucose starting as a substrate. No, they both do break up glucose. The difference is that this one is limiting oxygen. This one has enough oxygen. Only aerobic respiration produces carbon dioxide. That is true. Can anyone tell me the products of anaerobic respiration? Because this is important. They've actually highlighted this in your new syllabus. So very good, yeast. And yeast is an example of anaerobic respiration in which type of good lactic acid. So lactic acid is in animals. And yeast is in what? Oh, sorry, yeast plants. They release what? Ethanol. Very good. Ethanol. Perfect. Nice, guys. Okay, we're going to move on to 7. It says, what is a reason that Taq polymerase is a suitable enzyme for the use of the polymerase chain reaction PCR? We went over the PCR reaction in our weekend's crash course, and I remember a lot of students were struggling with it, and our lead tutor explained it very well. I was very happy with the comprehension, so hopefully you guys have also reviewed this very well in class. What should you think of when you think of Taq polymerase? It should be automatic. B, B, B. Good. Okay, a lot of you guys are saying B, which is perfect. Whenever you think of Taq polymerase, think of high temperatures. For this question, if you knew it, you knew it. If you didn't know it, it would have been hard to answer because you can't really use like any deductive thinking or try to connect specific topics. It's just how well you know PCR. So it is a bit of an annoying question to get on your exam, but we know that Taq polymerase is suitable for its function because of higher temperatures. Okay, let's do 8. It says a strand of DNA containing 4 codons with base sequences, this is our base sequence, mutates, changing the base sequence to this. What type of mutation has occurred? And this mutation is part of your new syllabus. So I would guess with like 70% certainty, you're going to get a specific question about mutation like this in your exam. So focus on learning these mutations. This is not HL, it's SL. But as an HL student, you can also answer these. If you guys would like for us to do an HL paper, just let us know and we can. Okay, a lot of you guys are saying B. Why are you guys saying B? What is the specific thing that is allowing you to say B? So we see that the mutation occurs in this section here. Try to use some active recall. So what is allowing you to say that it's B? Very good. Someone says ATG, GTA. G switched with A. Exactly. So I'm not sure what your name is, but you said switched. That's a very good word to use because it's a synonym as well for substitution. You substitute, you switch things. Okay, you replace them. And we see that here, they are substituted. So instead of going A, T, G, instead of going GTA, goes A, T, G. And that's why we know it's substitution. Insertion is when you insert a specific, so insert, use the wording to help you, insert or add a base. Here we don't see a base being added. We just add, we substitute. So that's why it's B. Delete. Okay. We delete a base. That's not what's happening here. Degeneracy. Can anyone think of another specific name, a more famous name for this specific mutation? It starts with S. Maybe you haven't heard of it. Maybe you have. Hopefully you have. Have you guys heard of silent mutations? So silent mutations are mutations that have no effect on our specific protein, because basically these silent mutations change the base, but this codon will still code for that specific amino acid, so nothing essentially ends up happening to our protein. So it's very good to have a silent mutation. Well, very good, because it's never good to have a mutation except if we're thinking of evolution. But a silent mutation is one of the best mutations because no effect occurs in our protein. Okay? One codon that codes for many amino acids. Perfect, exactly. All right, so I'm just going to check on the chat to make sure everyone's okay with this specific questions. Remember, guys, focus on mutations. Okay, I'm going to move on to question 9. It says the series of images shows 2 unicellular organisms found in ponds and lakes. Which functions of life are being shown in these images by organism X? What do we see? Someone already said D. Very good. I'm going to give other people... Okay, we have another D. D. Either A or D. Okay. I'll give you guys a hint to try to get a final answer. What's happening here? We see there's something here. There's another organism here. And what ends up happening is this, let's say this is Adam. And this is Philip. Okay, what do we see Adam doing? Adam engulfs Philip. Can we guess what's happening here? If it's engulfing it, very good, engulfing the thing, it's eating it up, right? So if it's eating it, it's for nutrition. And that's why our answer is D. Can we see why the answer is going to be D, guys? In a lot of the specimen papers, I think they also introduce lots of micrograph images. So make sure you understand your micrograph images very well, because I believe that there's also going to be lots of micrograph images in your exam. Okay? Can we see metabolism on the image? So think of metabolism as breaking down for any reactions that occur in the body. So you don't really see a chemical reaction occurring here. I mean, I guess you can, because you can say it's breaking it up to eat it. But it's more likely going to be nutrition. Okay? Why is it response? It's because it's responding. It's conscious that Philip is next to it. It first has to see it, recognize its food. It's responding to this, and therefore it's eating it. Okay? All right, good. What is a feature of phloem-sieve tube cells? Again, this is just something you'd have to know. If you don't know it, unfortunately, you wouldn't be able to know this question, but it's very famous. So phloem-sieve tube cells, what is a specific feature of these cells? B, very good, guys, no nucleus. Okay, good. So let me know if I'm going too fast, if I'm going too slow, if you want to speed up. I'll adapt to your guys' pace. Okay, let's have a look at question 11. It says, what can be identified in the electron micrograph? Here we see an electron micrograph. Remember, I said there's going to be lots of electron micrographs coming up. I think there will be as well in your May exams. So keep in mind, what do you guys identify? I'm just going to zoom in to help you here. And then I'll zoom back out so you can pick an answer. What do we see? Okay, I'm going to zoom out now. You can see a bunch of organelles. I'm going to circle the main one to help you guys. This is also a very famous one you should know. This one you should know as well. And these are the same. This should help you. Maybe you could have also identified it by this. Okay. It says plant cells with cell walls, nuclei, and chloroplasts. Plant cells with chloroplasts and a large central vacuole containing nuclei. Several prokaryotes with cell walls and cytoplasm. Animal cells with prominent mitochondria. What are we thinking? Very good. A lot of people are saying A. So yes, this is a plant cell with a cell wall. How do we know that this is a cell wall and just not a normal phospholipid membrane? It's because phospholipid membranes are very, very tiny. You wouldn't be able to see them. They wouldn't be this thick okay? And so whenever you see a thick wall surrounding our cell, we know that has to be a cell wall because phospholipids would barely show. Next, nuclei. The center, large thing in the center is a nucleus, okay? And we can see the DNA, the nucleosome inside. Chloroplasts, you should be able to recognize chloroplasts by their, just by the electron micrograph. You do need to be able to recognize them, and you can see them here because they have these specific structures in their cytoplasm, okay? It can't be B because large central vacuole containing the nuclei. The central vacuole should not be containing the nuclei. That's why we're going to cross this one out. Several prokaryotes with cell walls and cytoplasm. Okay, it could have been, but what do we see in the center here? We see a nucleus, so it can't be a prokaryote. And then it says animal cells with prominent mitochondria. Do animal cells have chloroplasts? No, we've identified already that these are chloroplasts. They're not mitochondria, so therefore it can't be D. That's why our final answer is A. How does everyone feel about this one? Someone said, is this from a past paper? It's a specimen paper. So we've done past papers before, and I think on the weekend we went live with a past paper, but because the new syllabus and the syllabus has changed, we're doing a specimen paper, so you guys are aware of little tweaks you have to focus on for the new syllabus. But we can do another past paper for you guys if you guys would like. Okay, next, question 12. You guys are doing very, very good. All right. 12 says the image shows a glycoprotein embedded in the phospholipid bilayer of a cell membrane. Which features of this glycoprotein can be deduced from the image? So here we see a glycoprotein. What can you guys see? Okay, I'm getting C's. C's, very good. Let's see actually if it's... So for your new syllabus, you need to know a little bit more about glycoproteins than you needed to know in the previous syllabus. So I would not be surprised as well if this showed up on your exam, because they're trying to, introduce new things, make sure students are aware, hey, this is a new syllabus, we're going to start introducing new things. You do need to know the functions of glycoproteins, and you need to know about the different variations of glycoproteins. So glycoprotein is a protein with glyco, so think of glyco, carbohydrates, carbohydrate chain attached to it, and that's what a glycoprotein is. It has very specific functions, so from attachment to cell recognition. There's a list of functions that you should know when it comes to glycoproteins, so make sure you're aware of those. Let's have a read of the questions. Lots of people are saying C, which is good. It is a peripheral protein that allows attachment to neighboring cells. It does allow attachment, we just named that as a function of it, but is it a peripheral protein? Remember, the question says what features of glycoprotein can be deduced from this image. Whenever IB is telling you to deduce something from the image, use the image. That's a little hint. Okay, so we can see here that this is not a peripheral protein. Why? Because although it does reach the surface and it can allow for attachment between neighboring cells, we can clearly see here that it goes all the way through the phospholipid bilayer. So it's not a peripheral protein. We can actually conclude that it's an integral protein. Okay, so that's why it's not going to be A. It says it's a channel protein that provides hydrophilic channels for carbohydrate transport. That is not a function of glycoproteins. It's not a channel protein. That is a function of proteins that you do need to know, but not specifically of glycoproteins. Next, C says it is an integral protein. That checks out. That may be involved in cell recognition. That also checks out because we know a function of integral proteins is cell recognition, and we can clearly see from this image we've deduced that is integral. That's why C is correct. Next says it's a transport protein that increases the permeability of membrane to glucose. No, it's not a transport protein. It's kind of like the channel or transport should not be a function of glycoproteins. Hopefully that made sense, guys, right? Someone says, what is the difference between integral and peripheral? So peripheral proteins just are on the surface of our cell. Integral go all the way through our membrane, okay? So this is one end of our phospholipid membrane. This is another end. Integral goes all the way through. Does that make sense? Hopefully that answered your question. Okay. What is a glycoprotein? I don't get it. So a glycoprotein is a protein, okay, specialized to have a glucose chain, sorry, not a glucose chain, a carbohydrate chain attached to it. What is the function of it? It has different functions that you need to know. One of them is attachment. One of them is specific cell-to-cell recognition. So how do cells recognize other cells? They recognize them through their glycoproteins, okay? And basically after your proteins are made in the endoplasmic reticulum, they go to the, what do they go to? They go to either the Golgi, they go to the Golgi, where they can fully change to have these attachments of carbohydrates. So if a protein's function is for attachment, cell recognition, and mainly to communicate between cell to cell, it will go to the Golgi to attach a carbohydrate chain, and this carbohydrate chain will help it for these functions. Does this make sense? Someone said, do you also do chem? We do also do chem, and we also have crash courses for chem. Perfect. Next, it says, how is the sodium and potassium ion ratio established during the resting potential of neuron activity? So we're thinking about the active transport. We're thinking of sodium-potassium pumps. Now, if you know about the sodium-potassium pump, you know that there's specific ions, which are sodium and potassium, that go in or go out. So what do we know? Do sodiums go in or do potassium go in? Which ones diffuse out? Someone said, what is the crash course? So our main goal here in Easy 7 is we provide crash courses. We provide 5-day crash courses where we review your whole 5-day syllabus. So for example, in day 1, we go through topics 1 to 3. On day 2, we go through topics 4 to 6, and so on and so on. So if you're SL, we review the whole syllabus in 3 days, and if you're HL, we review it in 5 days. Okay? We do have a chem crash course you can join online via our website. So you can just go on easy7ibsciences.com, or you can just follow the link in the bio, whatever you prefer. Okay, a lot of people saying it's B, it's A. Ooh, we're fighting. The girls are fighting in the chat. Okay, what are we thinking? This is just based on memory, so if you memorize it, you remember. Someone says, what if I didn't finish the syllabus at school yet? Is the crash course still helpful? It's very helpful. So the point of the crash course is to review every single syllabus point together. And it's mainly intended for those who haven't finished the syllabus, or those who are struggling with unhelpful teachers, or those who spend too much time making notes and don't want to spend time relearning everything by themselves. The crash course holds you responsible because you do show up and it's like a lesson. And it allows you to finish the syllabus and go above and beyond. So you might even finish it before the teacher gets a chance to finish it, which is nice. And then all you have to do is focus on practice questions, which is very helpful. Okay, people are saying B, people are saying A. We're still inconclusive. So the answer is B. So good if you said B. So, oh, why is it not annotating? B. So sodium ions go out and potassium goes in. Bonus points in the comments if anyone can say how many sodium and how many potassium go in. So how many sodium go out? 3, right? And how many? Very good. Someone said 3 and 2. And how many potassium go in? 2. Nice. You guys can just memorize it with a mnemonic. You can remember however you like. Perfect. Next says, what is a function of histones? So histones are what? What do they help with? They're specific proteins that allow for what? Wow, someone already said D. You guys are fast readers. D, good. Yay, all of you guys are saying D. So D are specific proteins that allow for the condensation of DNA, supercoiling of DNA. Perfect, guys. You guys are doing very well. So the supercoiling of DNA during binary fission in prokaryotes don't have histones. And prokaryotes duplicate or replicate via binary fission. We know that histones are not in prokaryotes, right? So it cannot be this because there's no there's no specific histone proteins in prokaryotes. That's why we can cross out A. Next, it says synthesis of protein. You could fall for this trap. Maybe they're thinking the student is confusing it with a ribosome because ribosomes help with the synthesis of proteins. No, they're the site of the synthesis of proteins. Next, it says formation of microtubules during mitosis. No, that's not the function. And it is for the condensation or supercoiling of DNA. That's why our answer is D. What is the specific organelle responsible for the formation of microtubules? Bonus points. Forcing you guys to do some active recall study. Someone said, why am I here? IB is controlling my life. It's self-reproduction process by prokaryotes, if I'm not wrong. Yes, exactly. Very good. Good job, guys. If you're helping each other out in the chat, it is a self-reproduction process by prokaryotes. Nice. So cytoskeleton. Have you guys heard of centrioles? So we're getting a lot of questions about what binary fission is. It's just how prokaryotes replicate. So eukaryotes replicate, not all eukaryotes, but how do animals replicate? So how does a human and like let's say a lion replicate? How do they have babies? Is the crash course the same as tutoring on the website? No, it's completely different. So tutoring, you get like one-on-one teaching. Crash course is like a live lesson with a lot more resources. You get taught the whole syllabus. Tutoring session is just 1 hour. Is the crash course online classes? You could think of it as online classes. Yes, it's on a Zoom call and yeah. Humans. Okay, hopefully this made sense, guys. I'm going to move on to question 15. It says the karyogram shows chromosome abnormality in human female. What could cause the abnormality to arise? So first of all, identify what the abnormality is, and then tell me what is the cause of that abnormality. And you should be very familiar with this abnormality. Hello, are you going to do physics? We don't have a physics crash course, but we can do physics past papers if you guys would like. Just let us know. Very good. Someone says trisomy 21. So if we take a look at chromosome 21, we see that there's an extra chromosome. And what is the syndrome name of trisomy 21? So you guys have said the scientific name. Very good. Does anyone know? Someone name dropped non-disjunction. Nice. Good, Andre. You said Down syndrome. So if we take a look at the answer options, we can actually find the answer. It says failure of homologous chromosomes to separate during gamete formation. Okay. Chromosomes 21 replicating after fertilization. That's not really a cause of Down syndrome or this extra chromosome. So the cause of an extra chromosome is something called non-disjunction, just like someone said in the chat. And this is because of the improper separation of chromosomes during meiosis. And can anyone tell me what phase of meiosis this occurs? Is it prophase, metaphase, anaphase, or telophase? Bonus points. What is the scientific name? It's trisomy 21. Trisomy 21. Why is it trisomy 21? Because you have 3 chromosomes. 3 chromosomes when you should only have 2 on chromosome 21. Very good. Someone says metaphase 1. Is it only metaphase 1 or can it also happen in metaphase 2? Let me know. A gene mutation of the mother's chromosome during cell division? No, Down syndrome is not caused by gene mutations. Polyploidy occurring during anaphase 1, meiosis in both parents? No. So polyploidy is like saying extra limbs, so no. All right. So someone said only metaphase 1. Actually, non-disjunction can occur in both metaphase 1 and metaphase 2, so be very careful. There's just different outcomes. Okay, hopefully that makes sense. So don't forget that it can occur in both metaphases because it's just the improper separation of chromosomes. And chromosomes separate in metaphase 1 and metaphase 2. So it can happen in both of them. All right? But for this specific example of trisomy, you just have to remember that it's a failure of homologous chromosomes separating during gamete formation. Okay, let's go to question 16. We're about halfway there. It says, which term describes the whole of genetic information of an organism? So, whole of genetic information of an organism. What are we thinking? And this is just a definition. What are we thinking? Okay, a lot of you guys are saying A, very good. So, a genome. And that is the whole of genetic information of an organism. So, in your syllabus, in the new syllabus, they've also focused on a different word. Can anyone tell me what proteome means? Because it showed up a lot in past papers, and it was highlighted in your new syllabus. So if a genome means whole of genetic information of an organism, what can a proteome mean? All of proteins. Very good, guys. So you just replace the genetic information with proteins. So make sure you remember that definition, because you could get a question like this, and it definitely has shown up in the past, and it's been a repetitive question, because what the IB does, it analyzes which questions that were not answered well by students, and then they give a statistic like, let's say 30% of students could only answer this, and then they repeat those questions to make sure that over time, this specific information is taught in school, okay? So it has been very hard, this question. Not a lot of people have been able to answer it in past papers. That's why they keep repeating it over and over again in hopes that the understanding will improve, okay? So make sure you remember what a genome is and what a proteome is. Okay, next it says, what occurs at gas exchange surfaces in lungs of mammals? So I'll give you a couple of seconds to think about it. Let me know. Someone says, do you maybe record the lives so we can watch all of them where we want to? Yeah, we do record them. Some are already posted on our YouTube. So if you'd like to watch more of these, we can post them on YouTube. We haven't recorded all the past ones, so I would suggest to follow. But from now on, we're going to post all of them on YouTube. So hopefully you guys can get a chance to relook at them. Okay, we're getting a lot of A's, D's, B. Is this biology? It is biology. Someone said all. Okay, so if you said D, you are correct. And there was only a couple of people who put D, so very good job, guys, if you put D. Okay, the answer is D. Let's have a look at the specific options. So it says gas diffuses across a moist surface. We do know that is in fact correct, right? Because we have pulmonary surfactant that is released by specific lung cells that allow the surface to be moist in the alveoli. So that is very important because it prevents the collapse of alveoli. If you're HL, you need to know what type of specific lung cell does that. So if you're HL in the chat, do some active recall and just tell me what specific lung cell. Okay? So if we know one is correct, I'm going to show you guys a little trick because not a lot of people think about this, but if one is correct, we can already eliminate which option? We could already eliminate C. Okay? Let's have a look at 2. It says concentration gradients are maintained by ventilation. That is also true, guys, right? Because when I breathe in, I take in oxygen, and then the oxygen, because I'm just going to draw my lungs here. These are my lungs. This is my blood, okay? Imagine I'm just zooming in, okay? In my lungs here. When I intake oxygen, the concentration of oxygen increases inside my alveoli, and then thanks to a concentration gradient, the oxygen diffuses into my blood cells. Why? Because there's low concentration of oxygen in the blood vessels, high concentration in the lung, and we know this diffusion goes from high concentration to low concentration, so we can maintain a sort of balance. So from high concentration, we go into the blood vessels, okay? And in the same way, CO2 is going to be a lot in the blood vessels, so CO2 is going to travel from the high concentration in the blood vessels to the low concentration in the lungs, and therefore, when we exhale, we release CO2. So we know that 2 is going to be correct. If 2 is correct, what can we cross out? B. Okay, so we're left with A and D. If this was a different question and you didn't know the answer, that would give you a 50-50% chance of answering correctly, and that's much better than having a 25% chance. So definitely use the cross-out method to help you with these types of questions. It will simplify everything in the long run, okay? Now we're left with just option 3. Now, is water lost? It is. So when you exhale, water is released in the form of water vapor. And a way to help you remember that is by thinking of when it's cold outside. You know, when there's like smoke coming out, or it sort of looks like smoke coming out of your breath, that's actually water vapor. Okay, so that's why our answer is going to be B. Someone said water moves up the xylem, so the next one is D. Oh yeah, good. You're moving ahead. Good. Water loss is a production of cellular respiration. Nice. And then, of course, the HLs said type 2, which is perfect. Okay. It is the type 2 pneumocytes that release the pulmonary surfactant. Okay. What is always a consequence of evaporation from mesophyll cells in leaves of a healthy plant? So here's my plant, and here's my leaf. What is always a consequence of water evaporating? So water leaving. how are the videos in the crash course different from the ones on YouTube? They're way different. So on YouTube, we only do past papers, okay? And in the crash course, you get a tutor who teaches you the whole syllabus. So think of reviewing all the content, even the tiny bits of content that your teacher didn't focus on or things that you struggle with. Every single syllabus point is covered in our crash course in just 5 days if you're HL, 3 days in SL. So it's very, very helpful. We also do have past paper working out in crash course as well. So you get both, the best of both worlds. Okay, a lot of people are saying D. So why is it D, guys? Do some active recall in the chat and tell me why it's D. Try to think of an answer. How would you type this up in your IB exam? It's very helpful to do this method as well. So our answer is D. So when water evaporates here from the leaf, again, there's a concentration gradient. We need water to be in that leaf. So what's going to happen is water is going to move up, okay, and travel to that leaf. It's going to evaporate, and then the process is a cycle of water moving up and evaporating. And this is beneficial because we require water for photosynthesis. Someone said negative pressure. Very good. So water will move up because we have that lack of water. Transpiration causes negative pressure, pulling the water to the side. Very good. So you guys have used that keyword or keywords that are required for your syllabus in the mark scheme. So also a very beneficial way to answer questions is by analyzing mark scheme, because it's really annoying when we know the content, we know what to say, but we phrase it wrong and we don't mention those keywords. So that's also very helpful because very helpful to look at the mark scheme, memorize the specific words you have to say to make sure you always get the mark. Okay. Next, it says what conveys messages from central nervous system to an endocrine gland. So what is the central nervous? What is the CNS? What is the endocrine gland? Try to reflect and think. Yay, you joined the crash course. Good. Very excited to see you next weekend. Where's the crash course? You can join via the link in the bio, or you can just search up Easy 7 IB Science. Is this GCSEs or A level? It's IB actually, but I think most of this content is also covered in A level. So if you want to stay for vibes, you can. Motor neurons. Very good. Someone said motor neurons. Nice. CNS is the brain and spinal cord. Very good. Interneurons. All right. Okay, this one's a bit iffy, so I'll give you guys a second. Okay, D. Nice, guys. A lot of you guys are saying D. Perfect. Motor neurons. So, is it B? Is it hormones? No, right? Because endocrines release hormones in the blood. And these hormones go to other receptors and cause a change in the body or cause a response in the body. But they do not convey messages from the central nervous to the endocrine. Very good. Okay. What about sensory neurons? What do sensory neurons do? Sorry, guys. What about interneurons? Receive stimuli. Very good. So it cannot be A. And then interneurons cannot be interneurons. Send signals to the spinal cord. Very good. Eve is on fire. I'm going to move on to question 20. It says, what cell is a component of the innate immune system? Why is it not interneurons? So interneurons connect the motor neurons and the sensory neurons. That's why. Okay, they're in between. That's why it's inter. Phagocyte, very good. B. Can you give a chance for other people as well? Phagocyte. Innate is immune. Very good, guys. So the primary immune response, the innate immune response, is how the body responds initially to specific pathogens. So, for example, coughing is part of the innate immune system. So if you cough, you're getting rid of all the any pathogens or any specific what's a good word to use? Any specific like things that are not supposed to be in your lungs, you can cough them out. So very good. Someone said innate is the natural response. Exactly. What are other things part of our innate immune system? And then lymphocytes, memory cells are all part of our secondary immune system. Mucus, good. So it's B. Broad range of pathogens. Exactly. So responding to bacteria, responding to viruses. Skin, nice. It's a barrier to prevent pathogens from entering. Good. You guys are doing amazing. Okay, we'll move on to the next one. How would the body respond to a rise above normal body temperature? So, above normal body temperature, what happens? So, positive and negative. Someone said, I don't understand positive and negative feedback. So, positive feedback is when something increases, it causes something else to increase. Negative is when something increases, it causes something else to decrease. Okay? So, let's say, we'll look at an example with temperature because the other one is hormones and that one's a bit too complicated, so I don't want to go into this specific detail on it, okay? But how would the body respond to a rise in normal body temperature? Let's have a look at the responses because it could also help us eliminate some answer options. Could the answer be shivering? When your body increases temperature, do you start to shiver? No, so it can't be this one. Okay. When your body rises above normal body temperature, do you spread your limbs to increase surface area? I mean, you can. I guess you can hold your armpits up to like let the air go through, but that's not really a response. Okay, so we're going to cross out this one. Increased secretion from sweat glands and vasodilation of skin blood vessels. So, vasodilation of skin blood vessels. When your body temperature gets too high, why do our blood vessels dilate? It's to try to release or dissipate some of that heat energy. Okay? And when it gets too cold, when your body gets too cold, your blood vessels start to shrink, right? To try to maintain that heat energy. So this is sounding correct. This is sounding correct. And then what is the type of feedback? Is it negative or positive? Shivering increases body temp. Very good, because you're shaking to try to generate heat. Move heat to the periphery. Very good. Vasoconstriction for cold. Negative. Exactly. Perfect, guys. And that's why our answer is C. Okay. You guys are doing very, very good. It says the first day of the menstrual cycle is when menstruation begins. What occurs around day 15 of the cycle? This is a difficult one. I always hated hormones when I was studying for my IB exam, and I never really got them until I got to med school. Okay. When is the crash course for bio? So we've already started the crash course for bio. This weekend we had day 1 and day 2, and last weekend. This weekend we're gonna have day 3 and day 4, and then the next weekend we're gonna have day 5. So we understand that not a lot of you can join because not a lot of you have days off yet, which is completely understandable. That's why we have them on the weekend, to ensure you can join us through the whole day. It's the whole day. We have lunch breaks, we have a bunch of breaks and specific, intervals for each topic, so you guys can follow along as best as you can. Will you do another crash course? So you can still join this crash course. And if you mean do you mean another topic? We also do have chemistry. Okay, a lot of you guys are saying A. Nice. Good job, guys. You guys are saying A. Let's walk through the answer options because I know with this question it's not very clear why it's A. How do you join the crash course? You can just join by searching up easy7ibsciences.com or joining the link in the bio. Okay, so I'm going to go through the other answer options first, and then I'm going to show you why it's A. Okay, so D says, or here, let's do the end of menstruation. So C is incorrect. Why? Because day 1 of our menstrual cycle, or our menstrual cycle is when menstruation begins. So if day 1 is when menstruation begins, does it make sense that the end of menstruation is at day 15? No, right? Because periods don't last 15 days. That's why we can cross out C. B says the placenta starts to secrete progesterone. When is the placenta formed, guys? Is it formed in a normal menstrual cycle, or is it formed when you have a baby growing inside you? It's formed when you have a baby growing inside you, right? So, and the menstrual cycle is always happening in a woman. It's not necessarily when the baby's growing. Perfect. So, no, that's why B is incorrect. And D says blood progesterone concentration reaches its maximum. I left this one to the end because I tried to eliminate the easier ones first. This one might have been a bit hard, but does anyone remember what day this is around? Good. Someone said day 21. Perfect, guys. It is at day 21. And what does this day 21? What does progesterone do? Can anyone tell me? What happens to the thickening of our uterus lining? And what does progesterone do? What effect does it have? It thickens the lining. Good. So progesterone helps grow that specific uterine lining to prepare for a baby. Okay, because when a baby implants, so when the fertilized egg implants in the uterus, you must have a thicker wall to maintain the baby. Okay, so blood progesterone reaches its maximum. Okay, to help maintain that wall, and eventually, when no baby binds, it shreds. Someone said progesterone is catabolic. Progesterone, no, so both hormones help with the growth and also the breakdown. You guys will pay for the April Fool jokes. I apologize, guys, for that. It was funny though. Was it not funny? Yes, drop in hormones will cause the shedding. Good. Is IB the equivalent of A level? I guess you can think of that like because A level's British. Yeah, similar to A level. Okay, but IB is like a 2-year curriculum. I know in A level you do some topics in year 1 and then some subjects in year 2. IB is like you do in both of the year 6 subjects. Okay, let's have a look at A. So why is A correct? Formation of the corpus luteum. When is the corpus luteum formed? When what? So this is our little egg. When the egg leaves our ovary after ovulation. Good, after ovulation. So when the egg leaves our ovary to enter the uterus through the fallopian tubes, we have a formation of the corpus luteum. And what day is that? The day of ovulation, or around ovulation. It's after ovulation, of course, because the egg has to leave, and that's around day 15. Okay. Next it says, what can lead to the emergence of analogous structures? I hated this topic. Hopefully you guys hate it as much as me. Okay, is this an HL past paper or SL? It's an SL past paper, but if you're HL, you can also do it. Eve also hates this topic. I agree, Eve. It's horrible. Ugh. Thinking about it makes me nauseous. This is horrible. Okay. It says analogous structures. Okay, what are... Oh, someone loves this topic. Okay, maybe you can tell us. Okay. If you love this topic, what's the answer? Analogous structure is divergent evolution. Okay. Is it divergent? Careful. It's divergent or the other one? I hate all topics. Valid. I'm pretty sure it's convergent. Very good. It's actually convergent evolution. What is an analogous structure? So an analogous structure is a similar feature or a similar structure, but is it a common or no common ancestor? I don't want to give you the answers so you can do some practice. No common. Very good. So you start at a no common ancestor and you have structures that are similar. What's a good example? You can think of bats. They have wings. Birds, they have wings. Butterflies, they have wings. Did they all originate from the same common ancestor? No, they didn't. But they have the same structures because they live in a specific habitat or an environment where they require to fly. Okay, so that's a good example of an analogous structure. And what is the meaning of divergent evolution and convergent? So divergent, this reminded me of Theo James, by the way. So what is the meaning of divergent? And after you've defined divergent, is this bio? Yes, it is. Tell me if you like the Divergent movies. Multiple into one. Good. Okay. So we can see that this cannot be divergent evolution. Okay, what is convergent evolution? Is species who have evolved what? Help, I don't know any of this and I've only got nuance of bio left. Join the crash course. You can learn the whole syllabus in 5 days. My name is Haneen. Okay, so convergent is something similar in all. So species who have evolved with similar structures but have unrelated, or they're unrelated in species, unrelated in common ancestor. That's why our answer is going to be B. Splits in the fundamental niche of species. What is a fundamental niche? This is part of your new syllabus, so I'm guessing this will also be in your M25 exams. Someone tell me what a fundamental niche is and what a realized niche is. Are we doing 30 questions today? We are. And then maybe if you guys would like tomorrow, we can do paper 1B. Just let me know what your preferences are. No idea, Queen. Oh no. What is a fundamental niche? You should know this. Or if you don't, you're going to know it. Okay, think of this example. So let's say a niche is what job this specific organism has within its environment, right? So what it eats, where it lives, and so on. Think of the example of what it eats because it's actually very helpful. So think of the example of this bird. This bird eats seeds, it eats worms, and it eats as well. Let's do another example: worms, seeds, and spiders. I don't know. It's just a fake example, but it eats seeds, worms, and spiders, okay? Let's say a snake comes along, and a snake likes eating these spiders and these worms. So the fundamental niche is the idea that this bird is supposed to eat all of these, okay? Its fundamental niche is its job normally that it would eat all of these. Its realized niche is what it's forced to do. So what is it actually forced to do in this specific environment? So because this snake comes along and it eats all the spiders and it eats all the worms, the bird is only going to be able to eat this specific seed, okay? So although it's supposed to, or it's allowed, its job within the organism is to eat all 3 options, its actual niche in this specific situation is only eating the seed. So why is this important? Because it helps scientists know the competition in the environment. It helps scientists know like how species have evolved. Nice. Eve said fundamental actual diet habits realize what they actually end up eating. Good, exactly. All the range is fundamental. Nice. Hopefully that makes sense, guys, because I know you said, many of you said they didn't know in the beginning. Did that help, guys? I'm going to move on. Okay, but that's why we know it has nothing to do with analogous structures. It's just, I think the way it would show up in your exam is probably in connection with competition. So they would talk about competition between specific species and then ask you which niche it would be, or it would just be a simple definition, define what this is. Because a lot of this topic, so topic 5 and topic 6 is a lot about like just say, what is the definition of this or define this, okay? All right. Yeah, you just started this unit. Okay, this is SL, by the way. I'm gonna move on to the next question. This is a hard one, but let's see if we can do it together. My screen is lagging. Hopefully you guys can still see my screen. Can you still see me? Okay, let's read this specific question. You can still see me. Okay, good. It says the number of hummingbird species in South America has increased continuously as shown in the graph. What could be a reason for the increase in the number of hummingbird species during the period labeled Z? So here we have an increase in hummingbird species. I'm going to give you a hint. Okay. Number of species. So not increase in just hummingbirds, but hummingbird species. So careful when you think about that. I'm going to quickly grab a charger for my laptop, guys. But go ahead and answer. I'll be back in 2 seconds. All right, what are we thinking? So I'm seeing a lot of B's and C's. Oh, D's as well. Oh, someone had this on their mock. How did your mock go, guys? Hopefully it went okay. Do I have to follow the course's schedule or can I get all the resources and do it at your own pace? You do not have to follow the course's schedule. So you get all the whole course is like recorded, and then after you can just open like the Crash Course Hub and then like rewatch the videos and use the resources in your own pace. That's why we release it like a lot of time before the actual IB exam, so you still have the time to do it. Oh, someone got a 7 in their mock. A 7, a 6. Mock was okay. Ooh, all of the mock was human physiology. All right, sorry guys, I got distracted. Okay, what is the answer? A lot of you said C's and D's. Let's have a look. So the hint for this question was we're focusing on species as a whole. The answer is C, but let's read the answer options. Okay, it says food and shelter are plentiful and there's a little intra-specific competition. What is the definition of intra-specific versus inter-specific? So intra-specific is competition within the species, right? Inter is between. So intra, we're talking about within the species. That does not explain why there is an increase in the number of species. Because if it was a decrease in interspecific competition, that would explain why there's an increase in the whole species. But here, we're just thinking about within. So that's why this is incorrect. This is a difficult question. That was actually just very rude of IB. I just realized. But yeah, it says closely related species can coexist indefinitely in the same niche. So if they closely related species can coexist indefinitely, is that even true? Can they coexist indefinitely in the same niche? Or what has to happen? No, very good. They cannot coexist because there's competition. So we're going to fight for the resources. We're going to fight for the space. We're going to fight for the food. We're going to fight for a lot of things. Okay? And that's why this statement as a fact is just incorrect. And it also does not tell us why there's an increase. Because actually there is competition and therefore you would assume that it would decrease, not increase. That's why B is incorrect. D says there are few limiting factors on the population size of hummingbirds. Okay. It doesn't really make sense because limiting factors are factors that would affect our population. And can there be few limiting factors? Someone says it's vague. It is very vague. And there's always limiting factors within our population. That's the true fact, right? There's always something limiting. Remember, oh, sorry, I was going to say remember in the crash course, but if you've studied this in class, maybe your teacher, remember how your teacher talked about when a population increases too much, what automatically happens? The population must drop, right? Because otherwise, if the populations kept increasing and increasing and increasing, there would be no resources on Earth. We have to give a period to allow resources to replenish, okay? That's why there's always limiting factors. So D is incorrect because it's vague and because it's just a statement we cannot say 100% it's true, okay? Someone said plateau phase, very good. Kind of plateau phase. Kind of. That's why C is correct. Okay? We're going to move on to 25. It says the preferred temperature ranges for 3 species of trout that are found together in freshwater lakes in North America are shown. Lake trout, rainbow trout, and brown trout. What can be deduced from this information? And I want you to use that specific trick we used in the previous question. So if you're locked in, you remember the trick of canceling out the options to help you. If you do chemistry, this is very helpful actually, because no, that was in my test today. Oh my god, what are the odds? Hopefully you answered correct in your test. What did you write in your test? You guys are doing very well, by the way. How regular are these lives? It depends on demand. So the more people comment on it, the more we usually do them. So if you guys are interested, we can even have one tomorrow. Just let me know that if you are. Tomorrow, maybe we can do the paper 1B, C, or D. Okay. You're interested? Okay, nice. What time tomorrow? What I can do is I can on the profile picture, I can like make it as an event so you can register the specific time because I would still have to look at my schedule and book a specific time. Okay. C, D. Okay, a lot of people are saying C and D. Let's look at the answer options to finalize. C, D. Okay, so we're still unsure. Yeah, we need these live daily. C. Okay, a lot of people are saying C. Good. Okay, so the answer is C. We're going to look at the answer options together. Hopefully, whoever had that in their test got that right. Okay, hopefully. Okay, it says the niches for all these species overlap. Okay, they do. Because remember, the niche we talked about is what they eat and what their specific function in the environment is, or what type of conditions they also like in their niche. And here we see that their temperatures, what happens to the temperatures? They kind of overlap, right? So we know some of the niches overlap because they live in the same temperatures. They're all trouts. So it makes sense that they sort of eat the same thing. That's why I is correct. So if I is correct, what can we cross out? We can cross out B. Okay. It says brown trout have the most specialized niche. Does this information tell us that? No, right? It does not tell us. It does not tell us which one's more specialized. We don't know. Maybe they wanted to trick you with the temperature because it has a lower temperature, lower temperature range. It has a more specialized niche. But no, this information does not tell us this. So 2 is incorrect. So we can cross out D. And so now you have a 50-50% chance. If you didn't know the answer, you could have just guessed from this point on. But it says lake trout can avoid competition by living in colder water. And yes, they can, because lake trout have a lower temperature range. So in order for them to, let's say, avoid the competition, because remember we said these niches overlap, to avoid having to fight for food or having to fight for spaces to live, they can live in colder environments. And that's why our answer is C, because both options 1 and 3 are correct. And if you remember, part of this unit is talking about how species will have a range of habitable specific environment they can live in. So it can be acidity, a range in which the acidity can change, a range in which the temperature can change, a range in which a lot of the specific factors within our environment can change, okay? So remember that in this question, you're analyzing the information, but you're also thinking about ranges and reflecting on what you learned about ranges in class, okay? So a similar question of this can show up. And the point of this question is to talk about the range in which a species can live. So in this case, that's why 3 is correct. Hopefully that made sense. How isn't the fact that its temperature range is large mean it doesn't have the most specialized niche? So specialized niche means it has a very specific niche. Okay, so if it has a larger range, that means its niche is not special because it can live in much larger, like different ranges of temperature. I hope that made sense. But if that didn't make sense, I can explain it again. But specialized is specific. So you have to live in specific temperature. So if something had, for example, can only live in 12 degrees Celsius water, okay, that's specialized. It can only specifically live in this. But since this is impossible, by the way, because all organisms have a range. But if something had a larger range, we can say it has more adaptive potential. Okay. Next, it says which row of table describes a type of nutrition. What can we say for this one? You guys have been doing very, very well. If you've been here from the beginning, good job, guys. Okay, I'm getting C, I'm getting A. A, okay, so I've gotten 2 A's and a couple of C's. C's. Okay, let's look at the answer option. So autotrophic means that they create their own food source. The first thing I think of is plants. How do the plants create their own food source? They create it through the source of energy of sunlight. Okay, correct. The source of carbon, organic molecules. What is that organic molecule? Someone do some active recall and tell me what that organic molecule is. Method of acquiring energy. Is it called respiration or is it called photosynthesis? We know that it's actually photosynthesis, so it can't be A. Okay, now heterotrophic. The source of energy is sunlight. Carbon dioxide is the source of carbon and photosynthesis. What's wrong about this? Someone said glucose. Very good. So the organic molecule is glucose. What are heterotrophs? Can someone tell me in the chat? Heterotrophs. What do we think of? Yeah, you guys are almost at 10K likes. They don't make their own food. Good. They gain their energy from other organisms. Nice. So the source of energy is not sunlight. It's definitely not carbon dioxide and photosynthesis. What's a mixotroph? Use the word mix to help you. They do a mix of both, right? So they do get from sunlight. They do get it from glucose. Their source of carbon is carbon dioxide because they can undergo photosynthesis and they can just eat glucose, right? They can just ingest things. So their source of energy is ingestion and photosynthesis because they can do both. That's why our answer is C. Now, consumer does not photosynthesize. That's why our answer cannot be D. Okay? Hopefully, this is making sense, everyone. We're almost done. We're on the last page. What could be helpful for the future lives is if you take a tally of all the questions you got right, and then maybe I could show you like a range of grade boundaries so you can grade yourself at the end of each paper, which I think would be helpful as well for you guys. All right, nice. Yay, guys, we reached 10K likes. Very proud of you. Are these lives recorded? They are, and we upload them on our YouTube. We haven't been uploading them in the past, but from now on, we're going to start uploading it because we've realized that it's very helpful for you guys. Okay, I'm getting D's. Herbivore, okay. Why is it herbivore? Why is it absolutely do that? Okay, nice. Well, I'll definitely tell the team to upload them on YouTube. Is this biology? It is, but we also do chemistry. So maybe at the end, I'm going to do like a little tally and let me know if you want to do tomorrow paper 1B or if you want to do chemistry. You can also do the chemistry specimen paper. We don't have a physics crash course, but we can do a physics past paper depending on demand because I also took physics in uni, not in IB, but I can also teach physics. Okay, we have secondary consumer. So with respect to the food chain shown in the image, what is the role of the locust? So this is our locust. Is it a secondary consumer or is it a primary consumer? It's primary, right? Okay, so it can't be A. Is it a parasite? No. Is it a chemoautotroph? No. It's a herbivore because it eats plants. Okay, nice, guys. All of you guys are saying primary. Perfect. Next, it says black walnut secretes the chemical juglone into the soil surrounding its roots. Juglone inhibits cell respiration in other species of plants. What does this example illustrate? I saw some of the crash course students here. We did talk about this in our crash course, or I think it's going to be part of the beginning of day 3, because it's in topic 6, and we finished on topic 6. Very good, allopathy, nice. And in the crash course slides, if you've had a look at crash course 6 slides, do you remember this specific example? So what's nice about analyzing the syllabus as a teacher or as a tutor is that you can actually choose specifically the examples that the IB is going to use in their exams. How can I do that? Is because I've read the syllabus and I've read the book and I can make these connections. This is an example that IB is going to use a lot for allopathy. So it's a bit hard to learn definition of things when we don't connect it to specific examples in the real world. Learning the example and the definition will help you. Because let's say you didn't remember what this is. Maybe you remember the example, okay? And specifically learning it as a sort of story form will help you remember the example. Very good. A lot of people are saying allopathy. So it's not parasite. It is part of topic 6. So allelopathy. So can someone, maybe if you've done the crash course, I want to see if you guys remember. But if not, that's okay. So allelopathy, what is that? It is when an organism releases a chemical which inhibits something else. Okay, that's the definition you need to know for your syllabus. The example that we said in the crash course, for example, is this black walnut tree. What does this tree do? It secretes juglone into the soil surrounding its roots, and that inhibits cell respiration in other species of plants so that this specific tree can continue to grow. So it's a form of reacting or preventing competition, okay? Because these trees are huge. They're very big, so they need a lot of space, okay? Does that help with the definition why the answer has to be C? So this is topic, it's topic 6 according to our crash course, because what we've done is we've taken like A 1.1, C 1.2, whatever, and we've conjoined them in topics. So this would be, if I remember correctly, 4 point something. I haven't memorized exactly the new syllabus. What I can do is after I finish these last 2 questions, I'll have a look and see which topic exactly it is for you. Okay, nice. It says, what is a density-independent limiting factor of a kangaroo? This is also part of your syllabus, new, and is actually showed up a lot in just the specimen papers. So remember what a density-independent limiting factor is. Can anyone tell me what it is? A, very good. Lots of people are saying A. Nice, guys. It is a forest fire. So what's a density-independent limiting factor? We've already kind of hinted at the fact that a limiting factor is something that limits our population size. So a density-independent, think of population density. So if something is density-dependent limiting factor, it depends on the population. Something that is density-independent, it does not depend on the population. So no matter the size of our population, this factor will affect it. So it doesn't matter if there's 2 animals in my population, okay? A forest fire is still going to limit this population. That's why A is correct. Predation depends on the population. So if there's high population, that means there's going to be high predator-prey relationship. There's going to be more predators, less prey, and so on. Okay, that's why this can be this one. Climate change does affect population because think about it as humans. An increase in population causes an increase in climate change. And that's why it's like a relationship between those two. That's why it can be climate change. And eutrophication. Can anyone tell me the definition of eutrophication? It's the excess, sorry, not excessive. Very good, nitrogen and water. So it's excess nitrogen and water, okay? And that's also dependent on the population, because the higher the population, the more of this we will see. Okay? Final question. You guys have been doing fantastic. It says, what is the outcome of natural selection? Do you think the M25 is going to have a lot of ecology units based on the specimen? I think it will. I think it will, not only based on the specimen, because a lot of the new syllabus is ecology. Okay? There is like a whole section about ecology that is just introduced and lots of definitions. I mean, in comparison to the M24 and all the other ones, yeah, there's just so much. I would definitely focus on that topic and focus on remembering definitions. Okay, a lot of people are saying A, evolution. Nice. So why is mutations wrong? Mutations cause natural selection. Why? Because mutations allow for variation, and this variation allows for natural selection. You cannot define natural selection as survival of the fittest in your mark scheme. Be very careful. If you say that, you will get no marks. but we can just use it in simpler terms now. So if something survives because it has beneficial features, that's what you have to say in your mark scheme, beneficial features, it's because these beneficial features are caused by mutation, and which causes variation, and that's why natural selection occurs. It's not an outcome of natural selection, but thanks to this that causes variation, we allow for natural selection. Reproductive isolation, that can't be correct. But evolution is an outcome of natural selection because survival of the fittest, the species that survive, the organisms that survive, will evolve, and that's why evolution occurs. Okay? For bio, I didn't and got a 6, never got a 6 ever. What did you do? Lost. Check right. Ooh, I'm not gonna read that comment. I don't want to get in trouble. Okay. How did you guys feel about this specimen paper? It was an SO1, but I would say it was. I'm gonna be mean and say that this is a tiny bit easier than what I expect the M25 is gonna be. But we have to keep in mind that it is a new syllabus, so I'm sure the IB will be nice in their specific past paper. All right, what I'm gonna do now is I'm just gonna show you. And if you have any questions, please stay and ask them. I'm happy to answer any study tips, anything generally about the IB. You guys did very, very well. Okay, a lot of you are asking about our crash courses. So you just go on easy7ibsciences.com. You go there and you have access to our crash courses. Oh, maybe I can show you the crash course hub. Hopefully I don't get in trouble for this. But I'll show you guys the crash course hub. This is members only access. Okay, so if you go on the crash course hub here, when are the IB exams and following the course for fun? IB exams they officially start in 22 days, but that doesn't start exactly with biology. So this is the crash course hub. I'm gonna go quick so I don't get in trouble. But the crash course hub, this is basically when you buy the crash course, what you see. Have you guys already covered the different micrographs? We have in the crash course, but you know what's nice? You go on day 1, you go on day 1 recording, and you get access to the day 1 recording. Okay, so you can cover it yourself. So you can zoom in, move here. Of course, this doesn't show you the chat. There's also an interactive chat where you can talk to the assistant tutor, you can talk to each other, you can ask the tutor to re-explain stuff. Here you can see an example of a past exam question. Teacher literally goes through every single syllabus point. So in day 1 and day 2, for example, here, because we've already done day 1 and day 2, you still get access to them. So you can purchase the crash course now, go on day 1 and day 2, watch them quickly, and then join us for day 3. You can even just purchase the crash course and not follow the days and just access the recordings, which is nice, right? All right, so here's, for example, is day 2. And then what do you get? So, for example, what are the resources? You get access to the slides. I can open a pair of slides for you guys to see. So this is an example of the slides. I'm not going to show too much. But here's the syllabus checklist. It shows how we've oriented the specific syllabus points because the new syllabus is very, very confusing. We've done it in specific topics, okay? And so then you can see a little bit about the notes. But these are just the notes, like you don't get the video. The video, you go in the recording. You get something called a resource pack. So a resource pack is really nice that you can annotate it while the teacher's talking. So what's annoying is when you're in class sometimes, you're taking in a lot of information. And the crash course, you're taking and reviewing a lot of information. So a way we allow students or help students remember this information is by having a resource pack. So rather than like having to retake your own notes and focus on, this is the wrong download. But basically to show you now, well, this is completely wrong. I think it's because it's on my iPad. Let's have a look here. There you go. Sorry, guys, I downloaded the Word document. But here's our resource pack. It has the syllabus checklist so you can check things off as you're learning them here. Okay. And then it also has, so let's say the first topic the teacher talks about, as we saw in the slides, was the emergent properties. What's nice is also in the resource pack, everything you need to be able to draw is listed. So learn how to draw a prokaryote, list, check it off. learn how to do everything. Check it off, okay? And then as the teacher is talking about emergent properties, you fill in the blanks. You define emergent properties. As there's a question, you answer the question, okay? So that you don't have to watch the crash course, take your own notes. You're creating your own notes. And of course, there's also notes available for you, okay? So that's nice as well. Is it not too summarized or what? It's actually not too summarized. I think it's really long. For 5 days, it's really long because we go from like 9 to 7. You don't have to be there all the time, but it's really not too summarized. We go through every single syllabus point. I would not call it summarized, but the crash courses for SL are 3 days, and then the last 2 days we cover HL content. So you can buy the full crash course, which is SL and HL. You can buy just the SL crash course, and then you can buy just the HL crash course. Do you need to label all structures in human physiology? You do. I didn't do well in my bio A. It will bring down my grade. No, it will not. It will not bring down your grade significantly. Do we do crash courses every year? Yeah, we have been doing them for around 4 years. We started in 2020. We opened the company in 2020. Yes, there's answers available. Okay, so someone just asked if there's answer available. I'm going to show you guys this lovely cheat sheet. So if you go on additional resources, you're not allowed to be doing this, but if you go to this specific resource, it gives you all the possible IB long answer questions you can get and the mark scheme for them. So what can they ask you about cell theory, what you need to say, how many marks you get, what you need to say about the eukaryotic cells, and the marks you get. Okay? And it's organized by topic, which is nice. All right. Is this video of this? Yeah, the video of this live will be on YouTube. So you have, what do you mean by all possible answers? So you have long response and you have short response questions. Your long response questions are usually very repetitive in IB. Okay. So what we've done is we've compiled the mark schemes. Where's the mark schemes available? It's available, like you mean this document, it's available in our crash course. Okay. What do you get? You get access to the specimen papers, you get access to how to solve MCQ. So actually, I created this document to also help you because one thing I did is when I was studying to get into med school, I did like exams that were specific, only multiple choice, and they were the worst things I've ever done in my life. But they taught me how to answer MCQs. So I created this document to also help you answer MCQs. Someone says, how much is the crash course? It depends. So for SL, it's a different price than HL. So SL is around 130. You get access to all the content and the videos and the recording and the live. And for the full one, it's 200, I believe, so only 70 more. And then HL, I don't remember the prices exactly, but yeah, so it's, if you think about it, it's £6 an hour. And usually the private tutoring is like £60 an hour. Not for us, but like usually outside tutoring is that. Someone just choose B or C. Yeah, this is actually factually incorrect. Oh, sorry, correct. I've analyzed and saw that either the answer is going to be B or C when in doubt. That's the IB's pattern, always. B or C. So whenever you're stuck. Damn, why is tutoring so expensive? Our tutoring is not expensive. I think it's actually the cheapest tutoring in IB. But IB tutoring is generally expensive, yeah, I know. But our tutoring is not. Especially questions 1, 2, 3. Wait, I'm lost. Especially those questions 1, 2, 3. Oh yeah, exactly. Those option questions, yeah. What time is the live usually? We don't have a specific live time, but we do. If you ever like go on our TikTok page, it usually says event, and it will let you know what that time is. Just because my schedule is not always decided every day. The ones with the answer options. Honestly, those might be my favorite because it helps. Like you help those questions with the answer options might be my favorite because they have like different options and you can like cross out A and B. And it's usually always a 50% chance that you're going to get it right. Oh, and I forgot to ask, tomorrow would you guys want to do a biology paper 1B or chemistry paper 1A? Let me know. Yeah, when in doubt, choose B or C. Because I've done, I think, all IB past exam questions since 2015 for both biology and chemistry, and it's B or C, I'm telling you. That's what I did for my exam when I was lost, not when I didn't. Okay, biology, biology, biology. Someone said chem. Okay, if we don't do chem tomorrow, wait, let me count. 1, 2, 3, 4, 5. Okay, so I think we're getting more biology. Tomorrow we'll do bio, and then maybe Friday we do chem. Okay, so tomorrow we'll do paper 1B. All right. Tomorrow we'll do paper 1B. And then Friday we'll do chemistry. If you guys want this to be a daily thing, please interact with us because it's really about demand. I don't mind having these lives every day, just as long as I know that, you guys want to have them. Okay. I love you too, guys. Bio isn't that hard to compare to chem, but you don't have to memorize as much for chem. So I guess it depends if you're good at memorizing or not. Guys, we need to lock in. Yes, lock in, guys. You worked or maybe you didn't work for 2 years, but imagine not working for 2 years. You just work now and you ace the exam. I would never stop bragging. Okay? Or the other option is you worked so hard for 2 years, you can't give up now. Okay? There's hope for any option that you did. Okay? Don't give up. You got this. All right. If everyone is content, I'm going to end the live. Have you completed? I have. If I start chem today, I don't know. Are you gonna lock in? If you lock in, maybe you can. If not, join the crash course. Thank you. Bye, guys. I'm gonna play a little video that summarizes the resources. Actually, maybe I'll play it tomorrow. But yes, join the crash course if you're interested. If you don't want to join the crash course, still follow us and we'll do more lives and help you guys in any way possible. If you want specific other free resources, maybe you want to do a live where we plan a study schedule, we can as well. Check the time of the live tomorrow. I'm sorry, guys. I really have to check my schedule to 100% make sure I can do it. Check the page of the TikTok and it will say event at this specific time, okay? At this point, is the crash course still worth it? Yes, it is. You still have access to everything, okay? So if you purchase the crash course, you have access to everything. Maybe you want to do Saturday where you, I don't know. Or maybe you purchase it and tomorrow you do all of day one. Like you put headphones in class and you don't pay attention to what the teacher's saying and you lock in on day one. Okay? What's nice is that there's recordings. And actually, I would say 60% of people who purchase the crash course because of the time zones don't actually attend the crash course, but watch the recordings themselves. And I know also a lot of M26 students have purchased the crash course so they can study with the teacher because you get access to this this crash course hub for 2 years, okay? So it's very helpful. Maybe if you have siblings, I don't know. It'd be helpful as well for them. Is the crash course in solving enough to get good grades? Yes, I promise. If you read our reviews, you will see it. I know a lot of, for example, chemistry. I want to say 90% of our students or 95% of a student at least went up 1 to 2 grades. So they had like 4 in their mocks and they got 6 in their final. So yes, definitely. Because one thing annoying about IB is that it's so much content and then you don't have time to do practice questions. And the way you get good grades is literally doing practice questions. Okay? And so if you waste time learning too much content, you're not doing enough practice questions, you're lost. Especially chemistry. Chemistry is so repetitive in past paper questions. They literally copy paste and they rearrange the options. So if you do lots of chemistry past papers, I'm telling you, you're going to enter the exam, you know answer is A. You know answer is B and you're going to move on. Okay? Or else, what's nice about chemistry is you can memorize how to answer the questions the way IB wants you to memorize them. Do you have tips for math? Practice questions. Okay, do a past paper, highlight the things you got wrong in math, and repeat doing those questions over and over again. Because again, math is also repetitive. If you're doing HL math, it's not the same advice, but if you're doing SL, it's very repetitive. If you're doing HL, it's a bit harder. For SL, try to focus on seeing which questions are repetitive and memorizing the step by step. For HL, try to see which difficult questions were asked and memorize them, because for HL, you have to learn a wider range of ways to answer things. You do need to lock in. Eve, you can do this. You were very interactive during this live. I'm sure you can lock in. You were doing amazing this live. Do you guys have an option for resources? Is the tutoring always included? No, so you can only buy the crash course. You can't buy the resources, unfortunately. Because the whole point of making you spend money is that you get the benefit of the videos, right? I feel like it wouldn't be worth it just the resources. My teacher dumped like 6 units in less than a month. I know, it's very, very annoying. If you want to learn ahead, use the crash course. Yes, you will. You will. You guys are going to dominate this new syllabus. Okay, I'm just going to answer if there's any tips for Bio-HL. For Bio-HL, the annoying thing about bio is that you have to learn the content, then do the practice questions. For chemistry, you can kind of memorize the questions, the question methods, and get away with it. But for bio, you unfortunately have to learn the content. So find a way you can learn the content very fast. If it's not a crash course, maybe you do like a YouTube videos, like watch a bunch of YouTube videos over and over again about the content, because the thing about videos is that it summarizes everything so nicely rather than reading and taking notes. Okay, so learn the content, then do the questions. For chemistry, if you want, you can start doing questions today and then highlight the ones you got wrong, learn why you got them wrong, and then do another past paper. And you'll see that over time, because the questions are so repetitive, you're going to ace it. Okay? All right, guys. I'm going to end the live now. We've been doing this for about 2 hours, and I do have to go, unfortunately. But I wish you the best of luck, and hopefully I will see you tomorrow. All right? But with chem, I just get stuck at every question. Doesn't matter. Keep doing them. Trust me, I was the same, and it gets better. Keep doing them. Like I did past papers up until 2015. So even if you keep getting stuck, power through. Okay? All right. Bye, guys. I hope this was helpful. I will try my best to have one tomorrow. If not tomorrow, then Friday. Okay? All right. Bye, guys.